like to start by really thanking the people in the laboratory that have been involved on this journey that we've taken for the last 20 years, trying to understand host microbe interactions. We've got a great team here in San Diego and a number of really important collaborators, and importantly, we've been very happy to be backed by the NIH and in particular the Atopic Dermatitis Research Network for the last 11 years, doing some of the clinical trials involved with this. And more recently, I'm a consultant and head of the SAB for Matricis Bioscience, which I think the industry academic partnerships that are now evolving in this field are making our capacity to progress quickly even greater. Just to set in framework the way we think about the skin microbiome, we've known for a long time that both the skin and the gut have very complex and comparable defense systems. This is from a review we wrote a few years ago. And it kind of, at a 1,000-foot level, illustrates the way we as immunologists and cell biologists have thought about skin host defense networks. And that is that there are specific molecules, specific receptors, specific cell types working together with canonical and non-canonical signaling pathways that generate function. So the challenge as immunologists and cell biologists have always been to understand exactly the mechanistic elements of this very defined group of cells that originate from the zygote, originate from the mammal to start with. Part of understanding this host microbe interaction has always been this old calculation of the surface area interface. And I'm sure everybody in this audience has, knows these numbers. The skin, classically, if you look it up on Wikipedia, and up until a couple of weeks ago, I thought of as about 2 meters squared. The GI tract, however, because of the multiple villi that take place on this tube, expands its absorptive capacity, giving a much larger surface area of 300 meters squared. So a priori, one may think that the host interface that we need to defend against as immunologists is much larger at the GI tract than at the skin. But on this camp ride from the NIH 2 weeks ago, I realized something. And that is, this is not the skin. We are not simply a sheet, which is what we've always used to calculate this surface area of interaction between our cells and the microbiome. There's something different about skin. And of course, this is obvious to us. We all know it. The skin is completely covered by follicles. Sometimes it's hair-bearing, sometimes it's not. We put a tremendous amount of evolutionary pressure on the skin of almost all mammals to develop these follicles, even when there's no hair coming out of them. You have to ask yourself why that is. And the calculations, kind of rough calculations, that the skin has about 5 million hair follicles. Even in my head, I have a lot of follicles, although not so much hair anymore. And the invagination of those follicles are about 3 to 4 millimeters deep. And if you take that into account as an epithelial surface area, which it most certainly is on the skin, that's the surface area of human skin. So, it's a rough calculation, but it's at least as large as the gut, perhaps 2 to 3 times larger. So what's the consequence of that? Well, to answer that question, we really need to know where microbes are interacting on these epithelial surfaces. So I'll start with a little kind of a simple experiment that we needed to ask ourselves if we were going to invest more time into understanding how the microbe might influence the human body. And that is to simply measure where do these bacteria interact with us. And to do that experiment, we took a number of human and animal skin samples, perfectly healthy skin, no broken barrier. And by laser capture microdissection, we're able to isolate different compartments of the skin, either the epidermis, the dermis, free of any appendage or connection to the surface, the hair follicle, which we just talked about, even fat, and then a number of controls. And from that tissue, these separate compartments, did a number of measurements to look at how many bacteria are there. And we were really surprised by some of this data. Direct your attention to the left first. Now this is just relative quantitative PCR looking at just 16S total. And the epidermis was set to 1. Each of these dots represents one individual. And as one would expect, there's more bacteria, at least more 16S DNA, can detect within the follicular compartment than within the epidermis. However, surprisingly and consistently, we see that all subjects we've looked at, in normal healthy skin, some microbial DNA was detected in dermis as well as in the adipose tissue below it. If one started looking at species-specific primers, such as a P. acnes 16S primer or a Staph epidermidis SOD-A primer, the same type of measurements could be seen. Lots of bacteria on the epidermis, lots more in the follicles, but quite a few, not as much as in the follicle, of course, but quite a few microbes were consistently detectable in dermis and adipose. And this was in comparison to a number of control tissues, just to look for contaminants. And when you start looking carefully, dermatopathologists have seen this for a long time, but kind of put it off as contaminants because it really couldn't be true. Here's just some immunostaining for Staph epidermidis. Sure, you see green Staph epidermidis within the superficial layer of the stratum corneum of the skin, but interestingly, at low numbers, one always sees Staph epidermidis in the dermis. And surprisingly, even in fat. And this is one adipocyte deep down in a normal, undamaged dermis. This is the fat body here, this big dark spot. And this is a RNA probe, so it's looking at RNA specifically within Staph epidermidis. And one can see that in the cytosol of the adipocyte, the bacteria can be taken up. That inspired actually a series of experiments which were published in Science this year, showing how the adipocyte is also an important part of the host defense system. How could this be? This seems impossible. Those of you in the derm field for a while realized that the classic size of molecules that could penetrate the stratum corneum is about 500 Daltons, much, 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 much smaller than a microbe. So how could the microbes be passing through a stratum corneum, an intact epidermis, and a basement membrane zone? Well, it's an active proteolytic dependent mechanism. This is a paper that was published this year from our lab where we took 3 different batches of Staph aureus and applied them to mouse skin. The first was a wild type strain of Staph aureus. The second was the same strain in which we knocked out all of the proteases that bacterium itself could produce. And the third was just take the same number of Staph aureus and kill them, heat kill them. If you apply them on the surface of the skin and do that laser capture microdissection of the epidermis, you see that the wild type bacteria and the protease null bacteria are present, relatively equal amounts. They're growing compared to the amount of DNA if we just apply those same CFU or CFU equivalents from the killed. However, if then one looks in the dermis, you see a dramatic difference. The wild type is again present in the dermis, consistent with the data I showed you from the earlier publication, but these live protease null mutants cannot enter the dermis, and nor do the wild type. So this tells us something about how it's possible that the microbes can be getting there. They're boring little holes through the epidermis. We have this giant surface area of 300 to 900 square meters of surface down all these hair follicles, and these microbes boring holes through the stratum corneum and the basement membrane zone at low levels, but giving it the potential to interact with live immunocytes and other cells below that rather impermeant superficial layer. So in part 1 of my talk, the conclusions are, first of all, I think the skin has been grossly underestimated as an interface for microbes and host interactions. And in terms of the surface area, this was an underestimation we were making even ourselves until the light bulb lit up a couple weeks ago. Microbes can penetrate the surface if they're alive, making this a rational discussion to say how microbes could be affecting human health if the live cells that need to detect them are protected by a stratum corneum across the surface. And then these physical interactions could be host and strain specific. For example, if strains have different abundances of proteases, perhaps they enter differently. If the host has a different barrier to that protease, it could influence that. So there's a lot of room for discovery in just this first part. Okay, so part 2, which is I think why we're here, and that is really trying to understand what the function of the skin microbiome. And This is a talk that's several hours long at this point. There's been a tremendous amount of preclinical data that has been accumulated over the years regarding how skin microbes might be influencing hosts in beneficial ways. Just to summarize the review we published a couple years ago, we like to think about it as three different concepts. First, some microbes have been suggested to be able to educate and prime the adaptive immune response, T cells, B cells, dendritic cells, and antibody production, autoimmune effects, and so on. Secondly, they may be able to enhance innate immunity, influence the way cells are producing antimicrobial peptides, influence the way the barrier is formed, influence neutrophil mast cell functions. And then lastly, some microbes might be protecting against other microbes. Are there good guys and bad guys? Do the good guys help us fight the bad guys? Maybe the easiest question of all to answer. The way this field is now, I think, being driven forward, especially in skin microbiology and skin microbiome research, is that there's obvious business opportunities for those of you interested in that aspect of things in the audience. Really, the top 5 dermatology conditions you've all heard of, they're very, very common: atopic dermatitis, rosacea, psoriasis, acne, and skin infections. There's at least 230 million people worldwide that suffer from these diseases. That's probably an underestimate. About 1,300 dermatology companies are attempting to address these, but most of these diseases are incompletely addressed right now. Perhaps the one exception to that right now is psoriasis, where we're finally getting a handle on some of the drug systems that can actually somewhat affect a 99 or 90% better treatment efficacy. And then these 5 skin diseases, which are still underexploited, represent at least a $12 billion prescription market and a $121 billion skincare market. So these kinds of numbers, I think, are inspiring various industrial groups to become interested in partnering with academics, trying to understand this riddle of what does the microbiome do. Okay, so for the rest of the talk, I'm going to focus and give you some brief information about atopic dermatitis, mostly because we see a lot of that in our clinics here at UCSD. We see about 80,000 dermatology patients a year through our network, and probably about 20% of them suffer from this in one form or another. It can be a debilitating disease. In some rare cases, it can be even fatal because of complications of superinfections. So it's a big deal. And there's a number of reasons for treatment of atopic dermatitis. Besides it being common, the genetic causes of atopic dermatitis are diverse. We know in general it appears to be one where there is a host defect in the barrier. So it can be a mutation in a gene which actually forms a barrier, or it might be a pool of mutations or environmental factors which makes that barrier less able to resist external stimuli. A big problem with atopic dermatitis is that it's frequently colonized by Staph aureus, and in fact, there's quite a bit of preclinical evidence to suggest that even at the lowest levels of colonization, atopic dermatitis drives this disease. So I'll talk more about that in a minute. We picked atopic dermatitis as a target not only because it's common, but because there's a long history of non-controversial information showing that atopic dermatitis is an excellent example of dysbiosis. This goes back to the 1970s, which by culture-based methods, Al Kligman and his group at that time just swabbed atopic patients and showed that they had very more bacteria on their lesional skin than non-atopics, and it was a predilection of colonization by Staph aureus in that study. And then more recently, first using, as this study did, 16S sequencing, and then more recently by shotgun metagenomics, showing that the community of organisms that can be isolated on individuals with atopic dermatitis is different than the community of organisms isolated from controls, and that community actually varies with disease flares. Typically, what happens when you get the rash, it gets worse, you get more staph species, particularly Staph aureus. So we and others have come up with a hypothesis for how the microbiome might be interacting with atopic dermatitis. If we believe the preliminary preclinical work that the microbiome has beneficial effects on the immune system, both in terms of the adaptive immune system and the innate immune system, then perhaps in situations where there's a genetic defect that leads to barrier defects in the individual, that barrier and that different environment then leads to dysbiosis, a community of organisms which does not have as much a beneficial effect on the barrier. That leads to some low level of inflammation and Th2 skewing, and then that drives further colonization by Staph aureus and this vicious cycle. And there's lots and lots of data to support all of these aspects of it, including the Staph aureus driving part. What we didn't have was great data on the dysbiosis driving this to start with. So that's our question. That's the question I'd like to address now, is does dysbiosis directly promote Staph aureus? And to address this question, we developed a series of different types of experiments, but I think this one was perhaps the most promising, in which we isolated individual colonies from normal subjects, from subjects with disease, isolated bacterial culturable colonies from lesional skin and non-lesional skin, and by isolating those colonies and putting them in a high throughput screening system with various culture conditions that were optimized to the particular question we were asking, we were able to screen thousands and thousands of colonies for biological activity. And the first biological activity that we wanted to screen was just the ability of one bacteria to kill another. So could a non-Staph aureus organism that is surviving on the surface of skin of patients with atopic dermatitis somehow influence the Staph aureus that's driving the disease? The results from that were really much more clear-cut than I expected them to be. These dots represent about 10,000 data analysis points. Each dot is an individual, so each individual is assayed a couple of hundred times for function of their microbiome. And what this shows is how frequently their community of organisms, bacterial organisms on the skin, would produce a soluble factor that would inhibit the growth of Staph aureus. And what we found in the non-atopic patients is that by far most of the culturable bacteria under these conditions secreted something that Staph aureus didn't like. It inhibited their growth. Not everybody, it was biology, but by far it was much greater. Interestingly, however, in the atopics, if you looked at their community of non-Staph aureus bacteria, those staphylococcal species were not able to inhibit Staph aureus. So somehow the community was shaped in a way that it now had a predominance of bacteria that said, hey, never mind, Staph aureus, go ahead and grow, you're not my problem. And this was even true in non-lesional skin, and we're breaking this out into individuals that had Staph aureus on them or patients that were culture negative for Staph aureus. And just in the next slide, you'll see this displayed a little bit differently. This is plotting now the amount of Staph aureus that we found on our subjects versus the percentage of other coagulase negative staphylococci that could inhibit Staph aureus. Up here, this is the group that has the high antimicrobial microbiome, mostly black dots, non-atopics. Down here is the low. And what's remarkable, was very, very remarkable to me for a clinical trial, is that we never saw a single individual that had a high amount of antimicrobial con S species that was also colonized by Staph aureus. So the association was perfect. You don't get Staph aureus if you have this function in your microbiome. And what was really remarkable, very, very rarely non-atopics have a little bit of Staph aureus growing. It's not a normal condition, it's not healthy, but they didn't have atopic dermatitis and it wasn't clinically apparent. And those people happened to pick up with a community of microbes that were low anti-Staph aureus activity. What does this mean? Well, this means that the microbiome might be important to resisting Staph aureus and treating atopic dermatitis. So if you turn to shotgun sequencing techniques and identify the species, we would hope that indeed we'd be able to use genetic approaches rather than this very labor-intensive functional screen on a species level to identify what's what. And although we see a very different composition of the community in the atopics and the non-atopics, unfortunately for the sequencing approach to this, Staph epidermidis, illustrated here with the yellow bar, can be frequently seen, of course, in non-atopics that are antimicrobial, but there's also Staph epidermidis strains now that are not functional, both the lesional and non-lesional. Similarly for Hominis and Warneri and the other species that comprise the normal human skin microbiome, you could find these species existing either in the functional group or in the non-functional group. So at the species level of identification, we're unable to distinguish between those that were beneficial and those that are not beneficial. So then we got to go back to the stuff we were doing 20 years ago, and that's sort of the brute force biochemistry and genetics to identify what the functional molecule is in these strains of coagulase negative bacteria. And this is an example of a zone of killing of Staph aureus by the target bacteria. This is a pigskin model showing that if we put this particular strain of Staph hominis A9, which we did whole genome sequencing and biochemical analysis and protein sequencing to identify the nature of these antibiotics, which surprisingly had never been described before, despite the fact this is found in about 60% of normal individuals. If you put that strain of Staph hominis on pigskin, you inhibit Staph aureus colonization of that model. None of these other strains of Staph hominis isolated from atopics were able to do that. In mouse, on a live mouse, you see the same thing. This strain of Staph hominis can inhibit Staph aureus. Other strains cannot. And if you apply it multiple times, this is just a single application, 7 days of application of Staph hominis can completely eliminate Staph aureus colonization of a mouse. So this looks pretty good. There's a nice commensal safe strain, we believe, Staph hominis. We designed a clinical trial. For sake of time, I won't go through it, but essentially the first trial was just to take our Staph hominis, put it in an over-the-counter cream that didn't kill it right away, and smear it on atopics on one arm, put the vehicle on the other arm, just put it on once, and go back the next day and see what it did. And we wrote a little NIH R21 grant for that, which we were funded, and we did this in a double-blind manner in an autologous approach where we harvested the subject's own antimicrobials and put it back on them. So these are the different strains, all unique, actually, antimicrobials. And what was really exciting to us in the 5 patients that we did this way, under our first IND, 100% of the patients responded with at least a log decrease from a single application of a commensal Staphylococcus strain. So we're very excited by that. So that's kind of the data right now. We're moving forward with larger studies, but I just wanted to leave you with my conclusions and then open this up for questions. We know atopic dermatitis has a large unmet need. There's abundant evidence that dysbiosis is driving this disease. The AD microbiome has a defect in anti-Staph aureus function, which I think is quite dramatic. And then rational screening has identified multiple activities. I've shown you one, but there's a lot of elements in the screening process that can help us move to the next level of understanding molecular-based, target-based canonical signaling events that can identify the specific strain or the specific genomic composition of that strain that would be beneficial for the target you're trying to approach. For example, novel kind of strains that kill Staph aureus, which I showed you. Antimicrobial function that's selective, and the ones I showed you have the added benefit as an antibiotic of not killing itself, so it maintains the normal community but kills the pathogen. Compounds that have anti-inflammatory action, we've published on this before. Compounds that can enhance the innate immunity. Factors that can inhibit toxin production by pathogens. And then ultimately strains that are safe, that are antibiotic sensitive, and are not biofilm formers. So applying them, we know a lot about them in case something should go wrong. The preclinical and phase 1 clinical trials have confirmed the safety, and I think they're showing great efficacy against Staph aureus, and we'll have to wait and see if this is a perhaps long-term cure for the disease. So I'll end just by reminding everybody this is a skin section about the skin. The skin immune system is complex, lots of skin's cells that participate in this. One of our cells that participate in this is the microbe, and I propose to you that we start thinking about certain members of the microbial community, not as foreigners, but as our own stem cells. These are multipotent, they're producing something that we need for health, and that they are regenerating. So they're really a type of skin cell. And with that, I'll thank you. Any questions?